Good afternoon, everyone. Thanks for being here. So for me, it's a pleasure to tell you more about what is the previews or the preliminary results of my thesis research. I'm a student of the University of Costa Rica, and I'm going to talk about the genetic diversity and clonal structure in wild populations of Vanilla odorata and Vanilla pompona in Costa Rica. So you've already heard a lot about Vanilla planifolia, and you already know that this is a highly valuable orchid. But as you heard before, it has a lot of problems because of its low genetic diversity, and it is due to several factors, but especially because most of the crops come from a single cultivar that is from Mexico, which is called Mansa. And the other factor is that in order to get the fruits, the flowers needs to be self-pollinated. So this happens in crops, but in the nature you can see that this is a highly phenotypic. It has high phenotypic diversity, both in the leaves. You can see different shapes, colors, and also in the flowers. So this phenotypic diversity may hold some genetic diversity. But why this is important? It is important because it can lead to adaptations to different climate changes and so on. So here in the neotropics, we have more or less 45 aromatic species. And here in Costa Rica, there are two of them, which are Vanilla odorata and Vanilla pompona. They are of special interest because they have this compound that is pretty valuable, the vanillin and some others. But also it is important to know the genetic diversity, which is not known until now, because they are on endangered category. So here you can see, like in these two plates, on the top, this is Vanilla pompona, and on the bottom is Vanilla odorata. We already know the pollinators of the pollinator of Vanilla pompona and the dispersers of both species. But what is the real rule in the genus is that the success of pollination is pretty low, or at least for Vanilla planifolia. And clonal propagation seems to be the rule. It's been seen that the natural reproduction is pretty low, ranging from 1 to 3% of flowers in nature, and only one fruit is seen per 1,000 flowers. And an individual of Vanilla planifolia can cover up to 0.2 hectares. So that our hypothesis was that populations of Vanilla odorata and Vanilla pompona have marked genetic structure due to clonal reproduction and reduced gene flow. So our aim is to describe the genetic diversity and the structure between populations and the fine scale genetic structure within populations. So according to the sampling, at the beginning we just went to Peninsula de Osa here because there are a lot of vanilla species over there. So there we could found three populations, or we collected individuals of three populations of Vanilla odorata and three populations of Vanilla pompona. But we wanted to increase the sampling. So we went throughout the Pacific region of Costa Rica, and we could find 5 more other populations of Vanilla pompona, but we couldn't find more of Vanilla odorata. It seems to be more restricted to the southern area. So here are the numbers. We collected 117 individuals of Vanilla pompona in 8 populations and 54 individuals of Vanilla odorata in 3 populations. So in our sampling, we consider an individual, a plant, which was attached to one for a fight. So in this picture, you're seeing 3 individuals. We mapped all individuals, and then we collected 1 to 2 leaves and stored them on silica gel. So this was the huge or time-consuming part of the project, which was to prove the transferability of microsatellites previously reported for this species. So as Dr. Crizzoni told you before, those are like African vanilla species. And for those species, they've already found some microsatellites. So I wanted to prove the transferability to these microsatellites to my vanilla pompona and vanilla odorata species of interest. And also, there are some microsatellites described for vanilla planifolia. I could transfer at the moment 18 microsatellites to vanilla pompona, but just 11 microsatellites are polymorphic. And in the case of vanilla odorata, I found 16 transferable microsatellites, but just 8 polymorphic. So I could continue with the process of genotyping. We use gene marker to genotype. Then for the genetic analysis, we calculate the number of alleles, which is A, number of effective alleles, heterozygosity, both observed and expected, inbreeding coefficient, and in terms of a structure between populations, we calculated the NACE GST, and we did a DAPC analysis. Then we wanted to know the fine scale genetic diversity so that we did a co-ancestry, we calculated the co-ancestry coefficient using GenAlex, and also we wanted to know like the clonal assignment and some other estimates using genotype. So the preliminary results that we have until now are that we found low genetic diversity for both species, slightly higher for vanilla pompona, but when we're talking about microsatellites, a high genetic diversity is about 0.6 and up, but here we just found 0.4. And a pretty important value is this, the number of alleles is pretty low. For microsatellites, a high number of alleles expected would be from 30 and up. So this low genetic diversity may be explained because of self-compatibility that has been proven for other species in the genus and because of the clonal growth seen in this species. Now talking more about the distribution of the genetic diversity in Costa Rica, we found that in the southern region of Costa Rica, we have the highest values of genetic diversity, and that in the Central Pacific, we got the lowest values of genetic diversity. And here it's important to take into account the effective population sizes. And if you can see in brackets, there are the number of individuals that we could collect there. So, sorry. So here, here you see that they are like a little bit more higher than in Garavito, which we only could collect 8 individuals. So the number of, or the size of the population is pretty important because small populations can suffer more the effect of genetic drift. Well, as I told you before, we develop a discriminant analysis of principal components. In this analysis, we want to look at like the different clusters that we find. Each clusters correspond to a population, so this is the population over here, and so on. What we found so far is that the populations cluster altogether here are those from the southern region, but I mean, the two of the Teraba Basin, which is located here, and one from Peninsula de Osa. Interestingly, we found that these two populations, also from the Peninsula de Osa, are not clustered together with these other populations. So we might see here like a significant structure value. And this is more evident when we do the DAPC in terms of regions. Here, I wanted to put the DAPC all over the map. Here is the Central Pacific region, and all the two populations that we collected here seems to be a little bit more related with the north, but the real pattern that we saw is that the two southern populations are clustered together. So we kind of see that the geographic distribution of population often influences their genetic structure. Okay, now talking more about the relationships or the kinship within the populations, which is called like a fine scale genetic structure, we have this graph. This graph tells you about the relatedness between the individuals in a population. So in the y-axis, you have the relatedness between individuals, and on the x-axis, you have the distance between individuals. You want that this line is up the confident intervals, so you can say that you found evidence of fine scale genetic structure. Here in the South Pacific, also we found that the individuals up to 7 meters are highly correlated. These are the values that you need to know. So for example, if the value is in 1, you have complete clones. If the value is 0.5, the relationship is too close, like full siblings or parent of offspring relationship. And if the relation is 0, you have unrelated individuals. So here we found that up to 7 meters, they are correlated, and after these 12 meters, let's say, the individuals they are not, don't have kinship anymore. Similar results were found in the South Pacific region, but in the Terava Basin, with an even higher value. So they are pretty related up to 12 meters. And in the Central Pacific, we found a little value, but still significant. Here I wanted to show you some coordinates maps of some populations to just describe these concepts. So this was one population in the South Pacific. Here you can see that there are 3 genets. So each genet is like a genotype. And in this case, each color represents that is shared by several individuals. So for example, the genet number 1 has 1, 2, 3, 4, 5, 6, 7, 8, 9 clones, and so on. And the evenness means that the genotypes are evenly distributed because all the clones have more or less the same number of individuals. In this particular population, we found that a clone could spread up to 20 meters. And in this other case, it's a little bit different. Here we have a high clonal diversity because we got unique genets. So these don't have clones, but the others do have. But so this clonal diversity is high because we have different types of genotypes. So what I want to say with these 2 clonal distribution maps is that the clonal structure varies between populations. And in this case, sorry, I didn't say, but a clone could spread up to 15 meters. Now talking a little bit more about Vanilla odorata, the genetic diversity was distributed this way. This is interesting because the highest genetic diversity was for a population with the least number of individual samples. But the least genetic diversity was for a population with a high number of individuals. So here happened the case in which it doesn't matter if you collect a lot of individuals, they may correspond to a single clone with 8 or more clones. Okay, so in terms of genetic structure between populations, we don't found genetic structure. All the 3 populations were highly similar, and this might be and this might be because of the distances that a clone could spread on the spaces they are found. Here we also found evidence of fine scale genetic structure up to 6 meters. The individuals were highly related. They were full sieves and so on. And here you're going to see the map of clonal distribution. Here we have unique genets, but we see that this is a really prevalent clone that could spread up to 85 meters. So this might be the explanation of why these populations in the South Pacific region are not structured, and the explanation of why these three populations are pretty similar. Still, we have to do more sampling over this region. So some key results of my thesis so far is that Vanilla planifolia shows interpopulation genetic structure, while in Vanilla odorata there is no interpopulation genetic structure. All populations can be considered one population in the peninsula de Osa. Both species showed evidence of clonal structure, and Vanilla pompona showed to have higher clonal genetic diversity. The thing of clonal genetic diversity is pretty important when you're going to the field, if you're interested in collecting vanillas or some other orchids, because this clonality can affect your estimations of diversity. And in this case, as I told you before, this might be due to effective population sizes. I mean, the effective population size can affect the genetic diversity of these orchids, which I found they were low. So because of that, we say that these two species are key species for conservation in the Pacific region. So some messages that I want to give you is that both species exhibit low genetic diversity. Both species display clonal structure, which lends them as vulnerable to population decline. The Southern Pacific region harbors the greatest genetic diversity in both species, and this may be related with the population sizes in this region. And this research holds significance for the conservation management of wild vanilla populations. So I want to thank the Universidad of Costa Rica, Lancaster Botanical Garden, Laboratorio de Ecología Molecular, and all these institutions for helping me with funding, and all the people that have helped me with the process of learning how to do all these different research things.
Thank you very much. And now we have time for questions.
Maria, beautiful work. I'm quite amazed of the distribution of the clones in that large area. Okay, those are quite large distance to have the movement or the growth of those vanillas, and they're probably just vegetative growth through time. And I was wondering if you had any idea how long it would take a plant to start growing and basically end up, how many meters away was the maximum that you've got?
85.
Yeah, exactly. Do you have any idea how long it would take to get that distance to grow?
I don't have any idea, but I know that vanilla are long-lived plants, so maybe a few years or more. I don't know if Adam has the information.
At the garden, they can grow at least 1 meter per year. Each stem, of course, so it can go. And so after it separates from the original plant, it can grow again and it starts making patches. So yeah, even faster. Yeah, possibly. Yeah. Even that.
10 meters. Right.
Do we have more questions? Oh, I can always count on you.
Alejandra, I have not understood how you correlate the elevation along the phorophyte with the genetic structure of the population. Which kind of relationship is between elevation and? I read in your slide, may be wrong, that up to 7 meters, or what it means exactly?
Okay, no. So you mean about the phorophyte?
Yeah.
Okay, no. We only consider in our sampling that one individual of each vanilla was going to be considered if it was attached to a single phorophyte. It doesn't matter the height of the phorophyte. Just if it was attached to a tree, it was the individual of each population.
Okay, thank you.
Okay.
Time for one more question. Do we have? I wanted to say that I don't see, but I do see.
Thank you, Alejandra, for your talk. I have a question for you. How do you plan to improve your sampling? You said, I need more samples. Do you think you can identify more individuals in these populations, or you plan to go to search for different populations? Or what are your plans?
We are planning to go back to Peninsula de Osa, find more populations because it's densely populated by vanilla species in there. So in order to improve the fine scale genetic structure analysis, it is better to have at least 100 individuals. So now I understand your question about the relatedness. I think this was that you was asking about. I can tell you later. But yes, we are planning to go back to Peninsula de Osa.
Okay, thank you very much.